Preventing dengue is a possibility, not a reality. Dengue, also known as breakbone fever, is a mosquito-borne tropical disease caused by the dengue virus. Symptoms include fever, headache, muscle and joint pains, and characteristic skin rash that is similar to measles. In a small proportion of cases, the disease develops into the life-threatening dengue hemorrhagic fever, resulting in bleeding, low levels of blood platelets and blood plasma leakage, or into dengue shock syndrome, where dangerously low blood pressure occurs. Asymptomatic in most of the cases. Typically, people infected with dengue virus are asymptomatic 80% or only have mild symptoms, such as an uncomplicated fever. So others have more severe illness, 5%, and in a smaller proportion is life-threatening. The incubation period time between exposure and onset of symptoms range from 3 to 14 days, but most often it is from 4 to 7 days. Signs and symptoms. The characteristic symptoms of dengue are sudden onset fever, headache, typically located behind the eyes, muscular and joint pains, and a rash. The alternative name of dengue, breakbone fever, comes from the associated muscle and joint pains. The course of infection is divided into three phases: febrile, critical, and recovery.
Transmitted by mosquitoes, dengue fever is a threat to almost half of the world's population. Dengue is a major public health problem, particularly in Latin America and Asia, where outbreaks occur regularly. When biting a person for its blood meal, an infected mosquito releases saliva containing the dengue virus. Once in the body, the virus infects the immune cells in the skin tissue. Enters the lymphatic system. The viral infection can then trigger a strong inflammatory reaction. During the incubation period, the virus first replicates locally. And then spreads into the bloodstream of the infected person. This is called viremia. For some patients, especially children, the infection may cause severe forms of the disease, including dengue hemorrhagic fever. The blood vessels become permeable, resulting in plasma leakage. Ultimately, dengue hemorrhagic fever requires intensive hospital care. When a mosquito bites a person who's already infected, it draws in the dengue virus contained in the blood. By biting another person, it can transmit the virus and spread the disease.
Dengue vaccine. The dengue vaccine field is facing numerous challenges. First, a viable dengue vaccine must be capable of protecting against disease caused by any of the four serotypes, a process that has been burdened by the absence of a validated animal model of disease or a well-characterized human infection model. Dengue vaccine challenge. The incomplete understanding of dengue immunopathology introduces risk into clinical development programs. Finally, the reliance on neutralizing antibody assays, which are notorious for inter-assay viability and cross-reactivity among serotypes, to generate immunologic endpoint data introduce error into data interpretation.
French pharmaceutical giant Sanofi Pasteur on Friday announced the publication in the medical journal The Lancet detailed results of its first landmark Phase 3 dengue vaccine efficacy study that could pave the way to combating the mosquito-borne virus that sickens millions every year. The late-stage trial involved 10,275 healthy children aged 2 to 14 across 5 countries in Asia, a region that accounts for over 2/3 of the mosquito-borne disease's global burden. Sanofi had already disclosed in April that its vaccine reduced the incidence of dengue fever by 56% in the Asian study, without giving details. The full findings were published online on Friday in The Lancet. Speaking ahead of the study's publication, Sanofi Pasteur's head of research and development, Dr. Nicholas Jackson, described dengue epidemics as a frightening event.
Vaccination of children with seropositive status produce high seroconversion rates and broad protein neutralizing antibody profiles. Despite such elicitation of antibody responses, why was there such disparity and inefficacy across the dengue serotypes? Could too much pre-existing immunity interfere with the serotype-specific vaccine response, leaving deficits in tetravalent efficacy? It is possible that the antibodies that were measured after vaccination were not all neutralizing but were a mixture of neutralizing and cross-reactive antibodies that were poorly functioning and potentially enhancing. Efficacy trial by Byler et al. Shows that we can protect populations from dengue disease and perhaps even reduce the proportion of patients with severe disease. Although the available results are not broadly generalizable across diverse populations, a foundation for additional studies has been laid.
Number 2. Dengue virus vaccine.
All it takes is one bite for dengue, a debilitating virus to be transmitted to humans by the bite of the Aedes aegypti mosquito. Half of the world's population is at risk of catching dengue. 50 to 100 million dengue infections worldwide every year, and the disease is fatal for 20,000 per year. The disease currently has no specific effective drug or approved vaccine. The ideal vaccine would have to prevent 4 different serotypes of the virus simultaneously. This virus can be found around the globe, most prevalent in tropical and less developed areas. The virus can also be found in highly developed countries. Local transmission has even occurred in Puerto Rico, the U.S. Virgin Islands, and parts of Florida and the Texas-Mexico border. More than 20 years in development, the world's first vaccine against dengue is showing positive effect on some variations of the disease. Clinical testing across Asia and Latin America show its greatest impact on the most severe forms of the disease. As the dengue threat increases, other vaccines are currently in development and undergoing clinical trials, including one that immunizes against all 4 dengue virus serotypes.